Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing University of Hyderabad PhD entrance examination for the subject Biochemistry conducted in the year 2020. It is for PhD admission 2021 session, and I will be discussing only the biology part. So coming to the first question. A lipase purified from 600 gram of bacterial pellet involved multiple steps. In the third step, a total of 100 ml carrying 300 mg of protein showed 72,000 units of activity. What is the specific activity of the lipase in this purification step? So here you have the formula for calculating the specific activity of lipase during purification. That is enzyme units divided by volume in microliter into protein concentration in milligram per ml divided by 1000. So this is protein concentration. My handwriting, I am extremely sorry about it. But this is the protein concentration. This represents protein concentration. Okay. So enzyme units they have given as 72,000 units of activity. So substitute 72,000 into 100 into 1000, that is they have given 100 ml. So converting it into 1000 microliter, 100 into 1000 into protein, that is 300 milligram, that is 300 milligram per ml. So per 100 ml. So per 100 ml divided by 1000 you will get 240 units per milligram. The next one, density gradient cesium chloride DNA density of cesium chloride gradient is. So this is the cesium chloride centrifugation tube, and it has got cesium chloride at different densities. That is from top to bottom it is increasing cesium chloride density concentration, that is gram per centimeter cube. So here you have 1.55, here you have 1.60, this is 1.65, 1.7 and 1.75. That is the solution, that is cesium chloride solution is less dense at the top and eventually gets denser towards the base. So if you add DNA into the cesium chloride solution, different density gradient density solution, it will migrate until it reaches its density. So here on the top you have got the proteins, then around 1.7 there will be DNA, and at the base you will find your RNA. So the answer is it has the density of cesium chloride, that is DNA density of cesium chloride is higher than protein and lower than RNA. So the answer here is option B. Then next one. Identify the correct statement. So cycloheximide inhibits mRNA synthesis while chloramphenicol inhibits protein synthesis in eukaryotes. Actually, cycloheximide it inhibits eukaryotic protein synthesis while chloramphenicol it inhibits prokaryotic protein synthesis. So the correct answer is option C. Then the next question. The next question, I have a doubt with the question itself, but they have given the answer key. So I am directly telling what the answer key they have given. Okay. If you have any doubts on this question, please let me know in the comment section. A protein migrates on SDS page as a single band, and if the same protein is subjected to native gel electrophoresis, you observe 3 bands at 20 kDa, 30 kDa and 50 kDa. What is the inference of the observation from 2 gels? So answer is protein has 3 subunits linked by disulfide bonds. That is why you get 3 bands, that is 20 kDa, 30 kDa and 50 kDa. And these subunits they are linked by disulfide bonds. Next one. Because my doubt was that in native gel I don't think they add anything that breaks the disulfide bonds, that you get a 3 bands. But in SDS page they add a reducing agent, that is which breaks the disulfide bonds, and you get 3 different bands. So the question, I think it should have been native gel of single band and SDS page with 3 bands. Then the protein has 3 subunits linked by disulfide bond would have been correct. But I don't know, it may be my mistake also. So if you have any doubt, you can just write it in the comment section, or you have your opinion about this question, then you can comment it there. Then next one. A cross between two homozygous recessive result in non-mutant progeny. This indicates. So a cross was made between two homozygous recessive alleles. Suppose it is AA and BB. Now this indicates that and they got what? They got non-mutant progeny. So if a homozygous recessive cross was carried out between two homozygous recessive parents, that is AA and BB, you should have got small a, small b. That is the usual Mendelian type of crossing. But what they got was they got non-mutant progeny. That is, they didn't get any mutated progeny. Instead, they might have got capital A, small b, and small a, capital B. Now, why this could have happened? This may be because that alleles of these mutant genes, they may be present in different genes. Alleles are or these alleles may be present on, that is small a and small b would have been present on different genes. So that may be the reason why they got non-mutant progeny. So answer is option A. Then next one. Given below are few statements about the Golgi complex. It synthesizes proteins, it does protein sorting to organelles, it does covalent modification on certain proteins, it has ribosomes on the surface of its membrane, it does packaging of protein into vesicle. Choose the combination that correctly represent the Golgi function. So Golgi, it synthesizes protein. Golgi doesn't synthesize protein. But what I have referred and I have found is that Golgi helps in protein synthesis. It is one of the key organelles required for the synthesis of proteins. The endoplasmic reticulum folds and alters the protein while the Golgi packages it for transportation. So it is involved in, it helps in protein synthesis. But protein synthesis doesn't take place in Golgi. So as per me, option one might be wrong, but they have given in the answer key that one is also correct. Okay, now coming to the others. It does protein sorting to organelles. That of course it does. It does covalent modification on certain proteins. Yes. It has ribosome on the surface of its membrane. It doesn't have ribosomes. Only endoplasmic reticulum is having ribosome. It does packaging of protein into vesicles. That is also correct. So the answer here as per me is 2, 3 and 5. But as per the answer key, 1 is also included and it is option D, that is 1, 2, 3 and 5 is correct. Okay. Now next one. What are the 3 basic steps of conventional PCR? So what does PCR do? It will denature, then anneal the primers and then extend. So the answer is option B, that is denature, anneal and extension. Then next one. Which one of the following dilutions can yield 52% sucrose solution when you are diluting 70% stock solution? So we will take each option and see which one is the answer. So if you take option A, so it is 10.4 ml stock plus 3.6 ml water. So if you take 10.4 ml from the stock, that is sucrose solution, and you add 3.6 ml water, you will get 70%. sorry, which one of the following you will get 52% sucrose solution when you are diluting from 70% stock solution. Okay, so we will see. So from 70%, how much you have to take to get 52% sucrose solution for 14 ml, because 10.4 plus 3.6 is 14. Okay, so that is 10.4 ml. So here the 10.4 ml stock is correct. Now we will see the other one, that is option B. If you add 11.2 and 3.8, you get 15 ml. So 70 into how much you have to take from 70 to get 52% concentration of for 15 ml, that is 11.14 ml. It is 11.2 here, so B is wrong. Similarly, you do for C and D also. You will find that A is the correct option. So answer here is option A. Then next one. Thymus and lymph node tissues from normal and RAG 1 or 2 knockout mice were extracted and subjected to FACS analysis to identify the population of double negatives, that is CD8- and CD4-. So double negatives, they are T cell progenitors, and they have not rearranged their TCR genes. TCR means T cell receptor genes, and they do not express proteins such as RAG1 and RAG2. And these cells lack CD4 and CD8 and are called double negatives. Okay, that is CD8- and CD4-. So they are called as double negatives. Then comes double positives, that is CD8 plus CD4 plus. And they have the beta chain of the T cell receptor being rearranged. Okay, and they begin to proliferate, and they also express RAG1 and RAG2 proteins for to rearrange their alpha chain genes also. So they are double positives, and then these double negatives, double positives, single positive, they are found at various stages of T cell differentiation, maturation, development, etc. And the single positive are either CD8 plus or CD4 minus. So double positive lymphocytes that express alpha and beta T cell receptor region CDR3 complex, they survive the thymic selection and develop into immature single positive CD4 plus monocytes or single positive CD8 plus thymocytes. So what they have told is, they have taken thymus and lymph node tissue from normal mice as well as the mice where RAG1 and 2 have been knocked out, and they have subjected it to FACS analysis to identify population of double negatives, double positives or single positives. Now T cells using fluorescein labeled anti-CD4 and rhodamine labeled anti-CD8 antibodies. Which of the following statements regarding the FACS observation will be correct? So the RAG1 and RAG2, I told you they are responsible for the T cell receptor gene rearrangement alpha and beta. So in RAG knockout mice, both the B cell and T cell are absent from the lymphoid organs because the cells with abnormal rearrangements, there is no RAG1 and 2 to help in the rearrangement of the T cell receptor region in developing T cells. So what will happen? These will be eliminated in vivo condition itself. So these T cells, they will be eliminated under in vivo condition. Okay. So we'll come to the first option. So thymus from normal mice will show all categories of T cells, but thymus from RAG1 to KO mice will not have single positive T cells. So thymus from normal mice, they will show all the categories of T cells. There you will find the double negatives, you will find double positives, you will find single positives also. But thymus from RAG1 or 2 knockout mice will not have single positive T cells. That is correct. Why? Because RAG1 and RAG2 knockout mice don't have that particular protein, that is RAG1 and RAG2, being synthesized for helping in the rearrangement of T cell receptor genes, that is alpha and beta. So these proteins are absent. So since there will be abnormal rearrangement of the T cell receptor region, this T cell population will be eliminated in vivo itself. So you will not find single positive T cells, and single positive T cells are formed at the final stage of T cell development. First you will have double negatives, double positives and single positives. So when the since RAG1 and 2 knockout mice don't have the RAG1 and 2 proteins, the double positives, that is CD8 plus and CD4 plus itself will be eliminated, because in this double positive stage the RAG1 and 2 are starting to express. So when the gene itself is not there for the expression of RAG1 and 2, naturally the double positives will be eliminated. So naturally single positives will not be formed. So the thymus from RAG1 to knockout mice will not have single positive T cells. Then so A is correct. Then next one. Thymus from RAG1 to knockout will not have double negatives and double positive T cells, but will have single positive T cells. So thymus from RAG1 and 2 knockout, they will not have single positive T cells. So that option is not correct. Then next one. Lymph node tissue from normal mice will not have double positive T cells, but lymph node tissue from RAG1 to knockout mice will have double negative T cell. Now lymph node, it is the secondary lymphoid organ and has population of mature lymphocytes. Okay, so mature lymphocytes means these double negatives and double positives won't be present in the lymph node tissue of a normal mice, because they have only mature population of lymphocytes. That is, they will have single positive CD4 8 and CD4 plus and CD4 8 plus lymphocytes, not immature ones, that is double negatives and double positives. So lymph node tissue from normal mice will have double positive T cell, and lymph node tissue from RAG1 to knockout mice will have only double negative T cells. They will have double negative T cells because they don't have the RAG1 and 2 proteins. So double positives will be eliminated from the population, and in case of double negatives, RAG1 and 2 proteins are not expressed. They are not necessary for CD8 and CD4 minus. So only the double negative T cells will be present in the lymph node tissue of RAG1 to knockout mice. So answer is option C. So both A and C are correct. Okay. So answer is option D, that is both A and C statements are correct. Then next one. N1 and N3 atoms of purine ring are contributed by which of the following? So it is by N1 aspartate and N3 glutamine. Answer is option D. Then next you have match the following. Match the contents between set 1, that is cell cycle protein, and set 2, its function. So the first one, that is APC cyclosome, it will be E3-UB ligase, that is active during mitosis. Then PRB, it will be a fourth option, that is cellular gatekeeper. And the third, P27, it will be the third option itself, cyclin dependent kinase inhibitor. And securin, it control metaphase to anaphase transition. by inhibiting separase. So answer is option B. Then next one. A mixture of compounds X and Y were separated by paper chromatography. During separation, X has travelled a distance of 6.8 centimeter. You can see the picture here. So it is X has travelled a distance from the bottom to about 6.8 centimeter. The compound Y moved 3.5 centimeter and is 5 centimeter away from the distance travelled by the solvent. So the compound Y, it has first moved 3.5 centimeters and then it is 5 centimeter away. So up till this region, that is this horizontal line, it represents till the region where the solvent has moved. So this is the whole length of the region where the solvent has moved. And the compound Y, it first moved 3.5 centimeter and it is 5 centimeter away from the distance travelled by the solvent. So it is this region. So what is the RF value of X? So you have to calculate first 3.5 plus 5 centimeter for Y. So distance travelled by the solute and distance travelled by the solvent. So solvent will be 3.5 plus 5 centimeter, that is 8.5. So you will get 6.8, that is distance travelled by X divided by 8.5, that is distance travelled by the solvent. It will be 0.8. So the answer here is option B. The next one. In a family, one parent has dominant phenotype and the other has a recessive phenotype, and the two offsprings have dominant phenotype. Which one of the following genotypes for parents with dominant phenotype can be deduced with certainty? So if you have, they say that one parent is dominant phenotype and the other is having recessive phenotype. So there can be two types of genotypes for parent with dominant phenotype. One is capital A small a and the other is capital A capital A. Whereas for the recessive phenotype, it will always be small a small a. So if it was capital A small a, then you will get offsprings capital A small a and a recessive offspring also. But they have told that two offsprings have dominant phenotype. So it will only be this particular phenotype for two offspring, the genotype for two offsprings, that is capital A small a. Now if you take the one parent to be dominant, that is it is having dominant genes for both the alleles, then capital A and small a, you will get capital A small a. So what they have asked is which one of the following genotypes for the parent with a dominant phenotype can be deduced with certainty. So the parent can be either homozygous or heterozygous dominant. So answer is option C. The next one. In ion exchange chromatography, often a salt gradient is used to elute the bound protein. This is because as the salts are neutral, they keep the protein active. A salt gradient allows the protein to ionize and bind specifically. At a certain ionic strength, the salt will compete with the protein for binding to the column. So that is the correct answer. That is why a salt gradient is used to elute the bound protein in case of ion exchange chromatography. Then next one. The mechanism regulating the decision between lysis and lysogeny for bacteriophage has been described as a genetic switch. The following 5 statements are made of this system. The C1 and CRO are transcribed in opposite direction. So that is correct. C1 and CRO genes are transcribed in opposite direction. The next one, the mRNA encoding C1 and CRO protein are synthesized using the same strand of DNA as template. No. The next one, of the 3 OR sites, that is operator sites in the genome, CRO has greater affinity for OR3. That is also correct. Then of the 3 OR sites in the genome, C1 has greater affinity for OR1. So that is also correct. Then E. Synthesis of C1 is stimulated by its binding to OR1 and OR2. So that is also correct. So A, C, D and E are correct, and you can find that it is in option D. A, C, D and E are correct. Then next one. You have amplified your favorite gene YFG of 0.5 kb in size using a forward primer containing a BAMH1 site followed by KPN1 site in 5' 3' direction and a reverse primer harboring BAMH1 site followed by SAL1 site in 5' 3' direction. You performed non-directional cloning of BAMH1 digested amplified product as the BAMH1 site of the expression vector PE, that is 3 kb. At the multiple cloning site of the vector, there are EcoR1 SAL1 unique sites flanking the BAMH1 site as shown in the figure. See here, EcoR1 SAL1 flanking BAMH1. After cloning of YFG in the PE vector, you have performed few restriction digestions and made the following observation. So what you have done is you have amplified your favorite gene YFG and it is 0.5 kb in size and you have placed it in the BAMH1 restriction site of this particular plasmid, that is PE. Now how is it placed? So YFG is 0.5 kb in size using a forward primer containing BAMH1 site followed by KPN1 site in the 5' 3' direction. So this is your YFG and this is the 5' 3' end of this particular circle. I have not completed the whole circle. That is because for your understanding I have not completed, only then I can mark 5' and 3'. So this portion of the circle, it will have BAMH1 KPN site along with, then followed by YFG, then and a reverse primer harboring BAMH1 site followed by SAL1 site in 5' to 3' direction. And this is the BAMH1 site, this is the SAL1 site, and this will be the 5' 3' direction for reverse primer. Now where is it placed? It is placed between EcoR1 and SAL1 site of the vector, that is PE, and the vector is around 3 kb in size. Now what they have done is they have conducted some restriction digestions and made the following observation. Now which one of the following statement best explains these observations? So first we will see. Digestion with BAMH1 yield a 3 kb and a 0.5 kb band. So here it will make a cut, that is here you have BAMH1, here you have BAMH1, it will release the insert which is 0.5 kb and this will be the vector backbone is 3 kb, so you will get a 0.5 kb band and a 3 kb band. So that's correct. Digestion with KPN1 yielded a 3.5 kb band. Now KPN1 has got only a single restriction site in the vector. So it will cut over there and you will get only a straightened plasmid. So it will be around 3.5 kb. So that is also correct. Then digestion with SAL1 yielded a 3.5 kb band. So that may not be correct because SAL1 there are two sites for SAL1 digestion. So you won't get a perfectly 3.5 kb band. Okay, now let's see the other one. A double digestion with KPN1 and SAL1 yielded a 3.5 kb band. So if you digest with KPN1 and SAL1, then you should have got a 0.5 kb and a 3 kb band because KPN1 is over here, then SAL1 is here and another SAL1 is here. So this 0.5 kb approximately would have been released and then another restriction site, that is between this region between the two SAL1, that also might have got released. So you would have got approximately 3 bands. So a double digestion with KPN1 and SAL1 yielded 3.5 kb band is not correct. So which of the following statement best explain this observation? The YFG insert did not get inserted into the vector. That is not correct because you are getting 0.5 kb band upon digestion with BAMH1. Then YFG insert got inserted into the vector in such a manner that the SAL1 site of the insert and SAL1 site of the vector were next to each other. It can't be because SAL1 BAMH1 site is over here and SAL1 and SAL1 are not next to each other. There is a BAMH1 site in between. Then KPN1 site of the insert got mutated. It didn't get mutated, but you got a perfect 0.5 kb band. The SAL1 site of the insert got mutated. That might be true. That is why double digestion with KPN1 and SAL1 didn't yield extra bands. It gave only one 3.5 kb band. So what we can make out from this observation is that the SAL1 site of the insert got mutated. Then next one, match the column 1 statements with best fit column 2 electron transport chain. Moves electrons from lipid soluble mobile electron carrier to water soluble mobile electron carrier. So that is complex 3. So 1C. Moves electron from substrate directly into the electron transport chain. So 2B, that is complex 2, contains 2 copper site. 3D, that is complex 4, irreversible inhibition in the presence of pyridine A. So 4A, that is complex 1. So answer here is option D, that is 1C, 2B, 3D, 4A. Then next one. A synthetic mRNA with the sequence, they have given the sequence, is translated in cell-free translation system. It will produce. So it will produce a single polypeptide. You see for the UAA sequences, then you will get a single polypeptide. Then it will produce a di and tripeptides. If you see for UAA regions, then you can count and see that it will produce di and tripeptides also. So answer is option A. Which one of the nucleotides mentioned below existed possibly early in the evolution? So it will be ATP that might have existed possibly early in the evolution. Then next one. Fusidic acid inhibits. Fusidic acid inhibits protein synthesis by binding to EFG-GDP, which results in the inhibition of both peptide translocation and ribosome disassembly. So the answer here is inhibits translocate activity of EFG. Answer is option C. Then how many supercoils can you see in the linear DNA carrying 360 base pair when it is unwound by 42 base pairs? You will see none because it is a linear DNA and it is not supercoiled. So answer is option D. Next one. Which one of the radioactive labels do you use to study DNA synthesis in cells? So you will naturally go for the alpha DTTP. Alpha phosphate group will be radioactively labeled in DNA synthesis studies. So answer will be alpha DTTP. Then next one. Which of the following structural features is not associated with all isotypes of antibodies? Presence of proline-rich hinge region, presence of carbohydrate groups on heavy chains, yes. Antiparallel beta-plated strands, yes. Region of hypervariability within the variable region of a light chain. That is also correct. Will be seen in all isotypes. Now before that, what are these isotypes? There are 5 isotypes in human antibodies, that is IgM, IgG, IgD, IgE and IgA. Now hinge region, that is proline rich hinge region, it is found in IgG, IgA and IgD, and it is not found in IgM and IgE. So this particular feature, it is not associated with all isotypes of antibodies. Next one. Which of the following statement is incorrect about oxygen-dependent microbicidal mechanisms used by activated neutrophils and macrophages? So TNF-alpha induces expression of inducible nitric oxide synthase in macrophages. So that's correct. Myeloperoxidases generate hypochlorous acid in neutrophils for antimicrobial activity. That is also correct. Superoxide dismutase generates reactive oxygen species to kill bacteria. Now, actually it is macrophages produce superoxide and reactive oxygen species to kill bacteria. During infection, some bacteria produce superoxide dismutase to protect them from being killed. So superoxide dismutase doesn't generate reactive oxygen species to kill bacteria. It is macrophages who produce superoxide and reactive oxygen species to kill bacteria, but some bacteria, what they do is they will produce superoxide dismutase to protect them from being killed. So option C is the incorrect statement. Next one. What general pathways can a eukaryotic cell use to synthesize nucleotides? So it can use salvage and de novo pathways. Answer is option B. Then next one. Which one of the following statement is true regarding the regulation of purine nucleotide biosynthesis? Adenylosuccinate synthase is inhibited by AMP only. So that itself is correct. So answer is option A. The next one. Which of the following amino acids are purely ketogenic? So lysine and leucine are the only purely ketogenic amino acid, as they are degraded into the precursors for ketone body synthesis, acetyl-CoA and acetoacetate. So answer is option C. Next one. Synthesis of 1 mole of cholesterol requires. So 1 mole of cholesterol requires 18 moles of acetyl-CoA, 36 moles of ATP and 16 moles of NADPH. So the answer here is option C, that is 18 moles of acetyl-CoA and 16 moles of NADPH. Answer is option C. Then histones are present in the nucleosomes as. They are found as 2 copies of each 4 histones. Answer is option B. The next one. The basic unit of chromatin organization is. It is nucleosome. Answer is option B. In which of the following organizational forms is the DNA present in eukaryotic metaphase nucleus? It is found as condensed chromosomes. Answer is option C. Then, which of the following RNA types has highest number of RNA modifications per 100 base pairs? So it is the tRNA type of RNA. Then next one. Which of the following best describes the process of genomic imprinting? This genomic imprinting, almost similar question have been asked just the previous year, that is 2019 paper. And I have explained there what is genomic imprinting, but you can refer to that. So the answer here is differential expression of allele depending on its parent of origin. Answer is option D. Then an enzyme catalyzed reaction was investigated in the presence and absence of an inhibitor. Michaelis-Menten plot of the data showed that maximum velocity did not change significantly when different concentration of inhibitor was used. Which type of inhibition is this? So for competitive inhibitor, Bmax remains the same as that of the normal enzyme, but the Km changes. So it is a type of competitive inhibition. Answer is option A. Then which of the following amino acid side chain can act as nucleophile in enzyme catalysis? So it is histidine amino acid. Answer is option B. Then which of the following is not correct about the function of the corresponding cofactor? Biotin helps in carboxylation. Yes. NAD plus as an oxidizing agent. Yes. Tetrahydrofolate transfers a 1 carbon unit. That is also correct. So what is left out is option D, that is pyridoxal phosphate help in transfer of alkyl groups. So pyridoxal phosphate, it has the ability to covalently bind the substrate and then to act as an electrophilic catalyst, thereby stabilizing different types of carb anionic reaction intermediates. It doesn't help in transfer of alkyl groups. Then next one. Which of the following transcription and translational mechanism are wrongly paired with its location? So translation, it takes place in the cytoplasm. Post-transcriptional modification, endoplasmic reticulum. Post-transcriptional modification of pre-mRNA such as capping, splicing, polyadenylation, it takes place in the nucleus, not in the endoplasmic reticulum. And after these modifications have been completed, the mature RNA molecule, it will be translocated into the cytoplasm where protein synthesis can take place. So answer here is option B. In endoplasmic reticulum, post-translational mechanism take place, not transcriptional modification. Then next one. In an experiment, alpha-amanitin was added with increasing concentration. Which below mentioned sequences correctly represents the sensitivity of RNA polymerases? So the various RNA polymerases, they are sensitive to alpha-amanitin. So the answer here is option B, that is Pol2 greater than Pol3, Pol1 is unaffected. So answer here is option B. The next one. Which one of the below mentioned processes involves metabolite sensing in non-coding portions of mRNA to control gene expression? So riboswitches is a regulatory segment of mRNA molecule that binds a small molecule, resulting in a change in production of the proteins encoded by the mRNA. That is called as riboswitch. So the answer here is option D, riboswitching. Then next one. Which one of the statements below best describes the function of helicase in RNA transcription? So helicase is involved in separation of the two DNA strands, giving access to polymerase enzyme. Answer is option B. Then next one. Sickle cell anemia is caused by a point mutation in hemoglobin beta chain at position 6. The point mutation is that is glutamic acid to valine. Answer is option A. Then which one of the following cellular processes is characterized by inflammatory form of lytic programmed cell death that occurs most frequently upon infection with intracellular pathogens and is likely to form part of the antimicrobial response? It is pyroptosis. Answer is option C. Then next one. Hemolytic anemia is caused due to the deficiency of. So the most common medical problem associated with glucose 6-phosphate dehydrogenase deficiency is hemolytic anemia. So the answer here is option A. Then which of the following is a ketohexose containing 3 asymmetric carbon atoms? Fructose, ribulose, tachyose, erythrose. So fructose is having these 3 asymmetric carbon atoms. Ribulose is having these 2 asymmetric carbon atoms, and erythrose also has 2 asymmetric carbon atoms. And stachyose, it is a tetrasaccharide consisting of galactose, glucose, fructose units. So the answer is they have asked for 3 asymmetric carbon atom, and it is fructose. Answer is option A. Then next one. Addition of inducer would not greatly affect the synthesis of beta-galactosidase in bacteria having the genotype. So mark the correct answer. Okay. So we will take the first case, Z minus Y plus A plus. So if you add inducer, it should not greatly affect with the synthesis of beta-galactosidase. So which genotype is it? They have asked. So Z minus Y plus A plus. Since Z minus, beta-galactose is not produced at all. So addition of inducer would not greatly affect with the synthesis of beta-galactosidase. So A can be one option. Then next one. I plus OC Z plus. So operator is constitutive. OC means constitutive operator, and repressor will not bind to this particular operator. It will synthesize galactosidase. So even if you add inducer, inducer will become bound to the repressor, but repressor already is not going and attaching to operator because it is mutated, it is constitutive operator. So beta galactosidase will be produced whether inducer is added or not. Then next one, I minus OC Z plus. Operator is constitutive here also. Repressor mutated, so it cannot bind to the operator. It will synthesize beta galactosidase. And next one, I plus O plus Z plus in the presence of glucose. So here the repressor remains bound to the operator. Operator is not mutated, it is O plus. So repressor bound to operator, and CAMP will not bind to the CAP. So RNA polymerase cannot bind and no transcription and translation of operon genes take place. Then next one. In the partial diploid, this is a partial diploid. This genotype, it is partial diploid. So in the partial diploid, I is mutated, operator constitutive, Z plus is active. Okay, this is the case with I minus OC and Z plus. In the other half of the partial diploid, it is I plus O plus Z plus. So inducer binds to repressor, RNA polymerase binds, and transcription and translation takes place. So beta galactosidase will be synthesized. So here only A, B, C are correct. But as per me, A, B, C and E are correct, and D and as per the answer key, I think they have given A, B, C, D are correct. But as per me, D cannot be correct because it is in the presence of glucose. So glucose is present means E. coli will naturally have a tendency to utilize glucose and not lactose. So answer is, as per me, answer is option D. A, B, C and E are correct. Okay, next question. Indicate the false statement. Gene knockout experiments involve replacement of wild type gene with mutant genes. Yeah, that's correct. Can be carried out across species boundaries. Yes. Are most interesting when they show no effect. So that is not the correct statement. It is the false statement. So answer is option C. May be helpful in determining whether open reading frames with no homology to other known genes encode functionally important proteins. That is also correct. Then next one. How much more acidic is a solution of pH 2 compared to a solution of pH 4? So the pH scale, it ranges from 0 to 14. And the pH scale, it is logarithmic, and as a result, logarithmic of 10 log you write. Okay, so it is a logarithmic value, and as a result each whole pH value below 7 is 10 times more acidic than the next higher value. That is, if it is pH 4, it is 10 times more acidic than pH 5 and 100 times more acidic than pH 6. So here they have asked for pH 2 compared to pH 4. So it is pH 2 is 100 times more acidic than pH 4. So the answer is option C. Then, given below are few statements about F2 progeny in a monohybrid cross. Identify the incorrect statement. So this is the monohybrid cross of Mendel, capital T with small t. You get the progeny. When you self it, you will get 1:2:1 genotypic ratio and 3:1 phenotypic ratio. So 3:1 is the expected phenotype ratio with one dominant and one recessive allele. So these are the dominants and this is the dominant allele and this one is the recessive allele. So that is correct. The next one, 1:2:1 is the expected genotypic ratio. So 1:2:1, that is the genotypic ratio. That's also correct. 2:1 is the expected ratio of heterozygous to homozygous recessive. 2:1 is the expected ratio of heterozygous to homozygous recessive. Okay, heterozygous, that is this one, capital T small t. There are 2 is to small t. So 2:1, that is the expected ratio of heterozygous to homozygous recessive. And 1:2 is the expected ratio of heterozygous to homozygous dominant. That is also 2:1 because it is heterozygous first and then the homozygous dominant. So that will also be 2:1, not 1:2. So the answer is option D, which is incorrect. Then next one. Which of the following statement is true for a quantitative trait? Example, crop yield. They are not amenable to molecular analysis. They cannot be inherited. They are multifactorial. Answer is option C. Then, the first electron acceptor of NADH dehydrogenase complex 1 of electron transport chain. So, the complex 1 is composed of FMN and an enzyme containing iron sulfur, FES. So the NADH molecule donates 2 electrons to an acceptor group, that is FMN. The FMN will get reduced to FMNH2, and from here the electron moves along a series of iron sulfur groups and then they are transferred to the coenzyme Q. So the first electron acceptor in complex 1 is FMN. Answer is option C. Then next one. Highest positive redox potential containing biological molecule. It is photosystem 1. Answer is option C. And next one. A receptor that has enzymatic activity other than ligand binding is. So it is insulin receptor. Answer is option C. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.